All right, everybody. So this week we're going to continue on talking about cloning stuff and lay out what you're going to do for the week to continue with the cloning project. So again, last week you should have started your PCRs using the fusion DNA polymerase to amplify the coding region of some genes of interest. And again, the coding region is the part from the of the messenger RNA that encodes protein, and ultimately that's what we're going to take and put into a plasmid. And again, when we designed primers, you could see how we incorporated different restriction sites into our primer sequences so that we had a gene specific part and a part that corresponded to our primers. And when we do PCR, when we have that, we have the restriction sites incorporated ultimately into our PCR products. And ultimately, we can do restriction digests and ligate those same restriction sites together into the multiple cloning site of the plasmid. All right, so let's think about what happens here as we have created a PCR product, okay? And so this long thing in the middle here is what I'm referring to as our main PCR product. But if you think about what's in that PCR tube when you get done your PCR reactions, right? We take a little bit of it, we run it on the gel, and we see the DNA, but there's other stuff in that PCR product, right? Like we have our Taq polymerases. There's a bunch of Taq polymerase floating around in there. We have a bunch of primers. Sometimes we see those primers on the gel. We have our DNA. We also have our DNTPs, our free pieces of DNA that haven't been incorporated into DNA strands. And so all of this stuff is floating around inside those tubes. And so if we want to do additional things to just this PCR product, we need to clean it up first. And so we have a protocol up on Moodle for DNA cleanup. And so it's kind of like a mini prep. We use columns and some buffers and stuff. And ultimately, when we get done that, we have a basically clean pieces of DNA in those tubes. So then what we do is we take that DNA and we digest it with our restriction enzymes of interest. And I'm showing you here based off of these DNA sequences, the restriction sites EcoR1 and KPN1, you're actually going to be using Hindi3 and BSHT1. But anyway, we set up our restriction digest, right? That cuts at the specific restriction site for those specific enzymes. And if you think about our DNA fragment, it's going to leave the main big center DNA fragment and then cut off these little ends, right? Where at the wherever the restriction site cuts, it's going to leave tiny little DNA fragments at the outer edges. But when we get done that, those fragments are still going to be in there. Our restriction enzymes are still going to be in there. And if we try to use this for anything, it's possible, but that those enzymes and little fragments could interfere. So what we do is we do another DNA cleanup to get rid of all of that stuff. And by the time we're done that second cleanup, we have basically a pure piece of DNA that has the free sticky ends hanging out each side, and we're ready to use that for a ligation. Really, we can call this an insert, right? We started by calling it a PCR product. By the time we get done doing stuff to it, we can call it an insert because we're going to be inserting it into the plasmid. Okay. Yeah. So then we basically need to do the same thing with the plasmids, right? So here's a crude diagram of our PEGFPN1 plasmid. You can see those restriction sites there at either end of the multiple cloning site. And so then we're going to take 2.5 micrograms of, or 2 micrograms of our plasmid DNA and digest it with those same restriction enzymes. In your case, you're going to be using Hindi3 and BSHT1. We're going to add those. It's going to cut the DNA specifically at either end of the multiple cloning site, right? If we look in here, Hindi3 AGE1 is the same thing as BSHT1. So we have Hindi3 and BSHT1. This little chunk is going to come out and it's going to leave all the rest of this plasmid DNA. Okay, and so that's what's shown here. This would represent our multiple cloning site, and this would represent the rest of the plasmid DNA. This is obviously not to scale. This is just my crude drawing here. So then we're sort of left with the same situation that we were here, right? We have some stuff in here that we don't necessarily want. So then we're going to do another cleanup, and the cleanup is going to get rid of the restriction enzymes. Cleanups generally don't do well with really small fragments of DNA, so it's going to lose most of the multiple cloning site there, and we should be left with fairly pure plasmid DNA with the sticky ends. And you'll notice that the sticky ends here are complementary to the sticky ends here. So ultimately, we can ligate our PCR product into the plasmid. Um, there's one more thing that we need to do to that plasmid DNA. So we're ultimately going to be doing a protocol called a ligation, where we take our insert and ligate it into our plasmid. Um, Sometimes if not all of the if the plasmid isn't digested with both enzymes, say that we had only really efficiently cut here, right? This would still be attached at this end. And if we had only cut right here, this piece of DNA could ligate back onto itself and create a circular plasmid. Um, and so in order to prevent things like that from happening, the ligation process requires phosphates to be at the 5' end of the DNA. Okay, so if you remember our DNA structure, right, we have a phosphate here. Remember 1, 2, 3, 4, 5' carbons. There's a phosphate at the 5' end. In order for ligation to work, the phosphate has to be there on at least one of the ends of the DNA. Um, and so if we do a ligation where we include another piece of DNA, um, that'll provide the phosphates enough for the ligation to work. So what we can do is remove the phosphates from our plasmid DNA, and we call that TSAP, thermosensitive alkaline phosphatase, right? Phosphatase just means it cuts phosphates off. And so what we do is we cleave the phosphates off of our DNA, off of our plasmid, I should say, right? And that prevents the plasmid from ligating back onto itself. And that's a key sort of way to prevent background in your cloning reactions. And so that's one of the things that I'm going to ask you to do this week. So you're going to be taking 2 micrograms of plasmid DNA, digesting it with your restriction enzymes, then doing a DNA cleanup, and then doing a TSAP protocol after that. And so hopefully by the end of the week, you're going to have your plasmid, or we can call this our vector, right? This just means it's a digested, cleaned up, PSAPed plasmid. So this is ready to go. Then you're also going to have your insert, which is our digested, cleaned up PCR product. So you're going to have 2 tubes, and you're going to have those nano dropped. We need to know the concentrations of DNA in each one of those tubes. Just be clear that, the last time we did a nano drop, you all got really nice curves, right? The where we saw the peak over 260 nanometers, and your concentrations were like 250 nanograms per microliter. Just be aware that your concentrations are going to be a lot less this time around. Okay, so your plasmid or vector fragment or cleaned up DNAs are probably going to be in the range of 20 to 25 nanograms per microliter. And your PCR products or inserts are probably going to be in the range of 5 nanograms per microliter. And you're probably not going to see very nice peaks there. It's possible you could see a nice peak, but it's probably going to be really low. That is perfectly fine. Don't freak out that the curves don't look as pretty as the first time we did this. Okay, so just to reiterate, you're going to take your Fusion PCR product from last week. If you got a nice clean PCR product, you can do all the rest of it on all the rest of the work on this PCR product. If you didn't get a nice clean PCR product, talk to me first before doing anything. Okay. So then you're going to do a cleanup on that. Then you're going to digest your cleaned up piece of DNA. We're calling this an insert now. So once you do your digest, you're going to do another cleanup on that insert and then do the nano drop on that. Okay. And at the same time you're doing your digest here, you can set up a digest of your plasmid DNA, right? So we told you to label your plasmid DNAs very clearly. You should have the concentrations written down in your lab notebook of what these are, or it should be written on the tube. If you don't, you can go and re-nano drop them. But you're going to set up the same restriction digest, right? And for all of Amy's things that we're cloning, the restriction enzymes are HindE3 and BSH21. Okay. So you're going to do your restriction digest on the plasmid, 2 micrograms of plasmid DNA. Do the DNA cleanup on the plasmid DNA. Do the nano drop on that cleaned up DNA. Then do the TSAP protocol on the vector DNA. Okay. You don't have to do a cleanup after the TSAP protocol. Part of the idea of it being temperature sensitive is that we can do the enzyme digestion on the DNA, and then we heat it up, and that kills the enzyme activity so that we don't have to do a cleanup after that. Okay, and so you're going to do your TSAP right at the end of everything there, and you're only doing the TSAP on the plasmid. Okay, we need those phosphates on the insert. Do not do the TSAP on the insert. We only want the TSAP protocol done on the plasmid. Okay, and so by the end of the week, you'll have 2 different pieces of DNA, and then next week we're gonna merge those pieces of DNA together. All right.